Right, so this is a little bit about the proposed 2-day workshop, which will contain balancing for pyrroles, folate, methylation and estrogen detoxification, and they're all connected, and it's stuff that I've developed in the last year. And so just this PowerPoint shows you what a pyrrole is. You can see it's a ring structure, 4 carbons and a nitrogen. And this is like a very simple pyrrole, but if it's got this ring in it that could have other little things hanging off it, but it's a pyrrole. And then you've got over here porphyrin. A porphyrin, you'll note, is actually 4 pyrroles made into a ring. And this may look familiar to you because the most important porphyrin in our body is heme, which is made into hemoglobin, but also heme is used as a cofactor for at least 100 enzymes. And so people that have the pyrrole actually have problems turning the pyrrole into porphyrins. And so what happens, you get this other factor called Mo factor being made, and it binds B6 and zinc, along with a couple of other, but mainly those ones, but a couple of other nutrients are important as well, and it depletes it out of the body. So people that have really serious pyrrole problems have zinc and B6 deficiencies and problems with that. So this is what happens. It's actually made from succinyl coenzyme A. Anyone ever seen succinyl coenzyme A before? Biochemistry? Yeah, it's one of the things in the Krebs cycle. So we have plenty of succinyl coenzyme A. So your Krebs cycles there being made in the mitochondria. So you get made to this D-aminolevulinic acid to porphyrobilinogen, and this is a pyrrole, okay? And from here, this hydroxymethylbilane is a porphyrin. So it's this enzyme here, 4 times PB deaminase, that makes the pyrrole into porphyrin, and it's this enzyme that people with pyrrole have a problem. And so if you don't make this, what happens is you get the Mo factor being formed. It'll bind your B6 and zinc, and whoosh, out it goes. And believe me, B6 normally we have plenty of because we absorb it through passive diffusion. But it does have to be, it can't be the pyridoxal phosphate. It actually has to take the phosphate group off, you absorb it, and then puts it back together. Because the pyridoxal phosphate is the active form of B6. So if you're taking it, often they have both forms, the pyridoxal and the pyridoxal phosphate. And you can see what happens if you continue along the way. You've got a series of enzymes working and producing this, uroporphyrinogen, coporphyrinogen, proto blah 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 blah, down to you get heme. Ferrochelatase actually puts the iron in there, so you make heme. And then if you're in a red blood cell, you get the globin chains put on there to get hemoglobin. But heme can also be used as a cofactor for enzymes. So, I mean, our red blood cells have 25% of our body cells, and we've got like thousands of heme molecules in each one. So we make tons of heme. So people who have pyrroles still make heme, because I'll tell you, if they didn't make heme, they would be dead. But you just don't make enough, and the problem is the enzymes. These 100 enzymes that are heme dependent are critical enzymes in, detoxification, steroid biosynthesis, degradation, and things like this. So there's a whole class, people that have the pyrrole problem often have toxicity problems. They can have problems. I was talking to a friend of mine in Germany, and her daughter has it really badly, and she just basically doesn't have a period. She's a 30-year-old woman, no period, because of the problems in making the steroid hormones, So it can be the impact of pyrrole disease can be huge, This is another pathway which shows you the chain here. Here you've got all the enzymes by enzyme number. A lot of enzymes actually have different names, but their number is standard. And so you can see here you've got this porphyrobilinogen, and then you've got this enzyme number 25161 that makes hydroxymethylbilane. It's this one here, 25161 is the problem in Pyrrole. Okay, and so it's often genetic and runs in families, and so that's one thing we want to look at with the 20, you know, if you get your DNA tested, say through 23andMe, is to look up this gene and to see if you actually have any problems with it, you know. I forget what it's called. So, and then you can see here you've got some little side tracks coming on, uroporphyrin, coporphyroporphyrin, and you can actually get this Pyrrole tested for the mauve factor, but you can actually get a test for all these other little ones too, which is interesting, because I think if you're testing some with Pyrrole, looking at all these should be tested as well, you know. And it goes up here and you get heme. And this 499911, that actually puts the iron in there. You can see you've got the iron in, it sticks it in the heme, and you've got the heme molecule with the iron in the centre. Okay, and so this then leads to all the issues that you have with especially the enzymes that are dependent on heme, and there's a whole class, a special class called Cytochrome P450s that are the ones that actually do the detoxification, and like, it's because I get a bit OCD with this, so then I looked up, you know, they said there's like 50 odd of these we have in humans. I found about 45 on gene cards, and so I wrote them all out, and I'm writing down what, you know, and it's interesting, this one detoxifies these, you know, they talk about what actually type of medications they take out, you know, and the same ones will start, and it's very interesting whilst talking about estrogen, is that estrogen's unique is it needs a Pyrrole dependent heme, a heme dependent enzyme, one of these Cytochrome P450s to break it down, and I'll show you that as an application, because it can be quite, you know, if you've got, for women, you know, it's only estrogen, none of the others, and especially in the target tissue like the breast and the uterus, you can actually have the wrong one, and that would actually lead to you having a high risk for breast cancer, you know. And I'll show you, actually it took me a lot to work this out, but finally got it, and I made it easy to understand flowchart. Methylation is the next thing we want to look at. Methylation and Pyrrole seem to go, you know, that if someone's got both of these, and especially this gene here called the MTHFR, it's a gene for an enzyme, that if you've got, because you've got 2 copies of it, and if you've got the 2 bad copies of it, you only methylate at 30%, and that's what the genetic website says. If you've got 1 good copy, 1 bad copy, you're methylating at 65%. If you've got 2, you methylate 2 good ones, you're doing 100% methylation. So, I mean, so this is a new balance I wrote last year, and you can see here, this is all this story is about getting folic acid in the right form so it can convert, or it can participate in the conversion of homocysteine into methionine. And see, people that have methylation problems will often have high homocysteine levels, right? And and this is this 5-methyl THF here. This is the activated form, and so you've got these different ways of coming in, but 5-methyl THF plus formaldehyde, plus homocysteine gives you methionine. So basically this methyl group, this enzyme here, MTR, is a methyltransferase. You can see I've got like the gene code, and they often quote gene codes, but here the class, the enzyme class 2.1.1. Any enzyme that's 2.1.1 is a methyltransferase. That means it does methylation, and there's about over 200 on the enzyme database now. I went through each one in my OCD way I do, and found about 30 of them are in humans. Some of them are like in fungus and bacteria, because they have their own enzymes that methylate too, and the enzyme database includes all enzymes in all species, right? So I had to go through each one, and I plucked out all the human ones, and then I categorised them into different, because you know, you might have like 8 that methylate RNA or something like this, or 3 that do DNA and stuff like this. And so, and this gives this SAM, which is the SAM, or the S-adenosyl methionine, and that's that methionine gets converted to that, and that actually donates the methyl group in about, I think out of the 30 odd, it's like 26 need SAM. There's like 4 or so that don't need it. So this whole pathway of getting methionine from homocysteine is critical, and you need 5-methyl-THF. And to get that, you've got, you know, you can get like different pathways. Folic acid gets made to tetrahydrofolate. THF means tetrahydrofolate. Then you've got 5,10 methylene tetrahydrofolate, then 5-methyl tetrahydrofolate. You can see there's different pathways. All of this step from methylene to 5,10 methylene THF to 5-methyl THF, this is where the MTHFR comes into it. Cofactors FAD, which is an activated form of B2, okay? And so you can see here that people, and it's this MTHFR gene is the problem. So it's you can actually supplement this, but it's actually, you know, you would need to talk to a naturopath, you know. So you can see here you've got these different ways coming in. And when it donates, so what's interesting is when it donates the 5-methyl THF, you get, you know, the methyl group goes to the homocysteine, you get methionine, but you've got the tetrahydrofolate, and that can go back in and be recycled in the whole process. So it recycles around, okay? Now, so this is interesting. So here you've got the folic acid. Now you've got this other supplementary pathway here, which is which is called the TMG or betaine DMG pathway, which works side by side. Okay, and so this trimethylglycine plus homocysteine, it's got 3 methyl groups. It can donate one of its methyl groups and get methionine, and so that actually bypasses the need for folic acid and the MTHFR. Again, if you've got the two dodgy MTHFRs, you should talk to your naturopath about supplementing. But then the DMG can actually donate them. You get it can donate sarcosine and donate the methyl group into the 5-DHF, and you start getting into this pathway here where you get the 5-methyl THF over here. But these ones need the MTHFR, okay? And so then with the SAM cycle, this is basically what happens. Then the S-adenosyl methionine, plus this is when you're methylating DNA, because methylation of DNA actually switches off genes. It's an epigenetic factor, but it's also, you know, later on I realised, you know, where you've got to actually address all the 30-odd methylation enzymes that exist in humans, okay, and about 26 of them use this SAM cycle. So you get the DNA with the methylated cytosine. The cytosine's actually methylated, and that'll switch the gene off nearby, and that gives you S-adenosyl homocysteine, which then again gets converted to homocysteine and plus a cysteine, and homocysteine can come back here to the methylation pathways, or can be broken down into cysteine, which is then important for making glutathione, because cysteine availability limits your ability to make glutathione, and glutathione's important antioxidant, free radical scavenger, so it's very important for cellular protection. So that's methylation. And then the last thing I want to show you is the estrogen, and this is pretty fascinating. And so this is estradiol is the main estrogen in non-pregnant fertile women. And you can see this CYP, all of these are cytochrome P450s. So in the liver, when you break down estradiol, you've got, and you'll notice the enzyme number's the same. So at first glance you say, oh, it's the same enzyme. But this enzyme's got a number of genes. It's got the same number because it does the same thing, but it's a different protein with a different shape and work on different things, right? And so if you know, in the liver you can produce 3. So over here, if you've got the 16-OH estradiol, that'll then break, you know, and this in detoxification, you've got 2 steps. First step is modification, so this is doing the modification, and that's adding an OH group. And you can see 16, 2 and 4, you know, that's the number of the carbon. It's just putting the OH group on a different carbon, and that makes all the difference. So then you've got the conjugation. Here you've got class 2 enzymes, which are transferases, so they're always the conjugation are a class 2 enzyme. And this goes down here, this one gets stored in the liver, this one's here secreted in the liver and kidneys. Okay, and same over here, but over here the conjugation is done by COMT, which is a methyltransferase. So COMT's known, it breaks down dopamine and it breaks down norepinephrine. And it's actually the warrior gene. I don't know if you've ever heard the warrior gene. So the people who've got the warrior gene, their COMT is slower than the warriors. So if you're a female and you've got the warrior gene, you're going to break down these estrogen products slower. So down here, no drama. It may break down a little slower, but you get it out in the liver. But over here in the target tissues, this is where you've got the interesting thing. You'll notice that the enzymes, you've got the CYP numbers are different. And so in the target tissues, you've got breast, uterus and ovaries. And you've got 2, the CYP1A1 and CYP1B1. So this is the 1A1 gives you the 2 and the 1B1 gives you the 4. And this is the dangerous form of estrogen. It creates reactive oxygen cell damage and carcinogenic. So if you're a female and say if this enzyme's not this gene's not good, and you're relying on this gene to break down the estradiol in your breast, uterus, ovaries, etc., right? Then all of a sudden you're starting to get risk for cancer, and especially if you've got the slow COMT. Then it's hanging around longer, and you know, and so then you've got this. It comes down here, and you secrete it. So this is pretty interesting, you know, and it includes pyrrole and methylation. And when you look at the actual production of all the steroid hormones, they've all got pyrrole-dependent enzymes involved in the production of them as well. So, but often the methyltransferases are used as a form of detoxification. This is actually a detox pathway, you know. So these are the topics of this course. So you've got pyrrole, folic acid, methylation, estrogen, detoxification. A lot of formatting in it, and I'm pulling bits out of 2 courses to put it together to make it a general interest 2-day course. So it's wham bam, thank you, and you know, you can have it all quickly and very effectively use it. And I will give you in the course all the different methylation, forms of methylation. I broke them down into different parts. You know, there's things like, you know, making of melatonin, breaking down of neurotransmitters. Histamine, you know, is a problem. dopamine, norepinephrine, serotonin. Even I found the pathway for making DMT, actually found the pathway in things. And again, making DMT, dimethyltryptamine, you need to add 2 methyl groups, so that's methylation. You can actually go to 23andMe, get your DNA tested, and then run it through Genetic Genie and get your methylation profile and your detox profile, because the detox profile has all of these enzymes, and your methylation profile has this, and MTHFR and all that. And so then you'll be able to look at what if you've got any problems, and then we can get into balancing your actual SNPs or single nucleotide polymorphisms, or it's what allele you have,